Okay. Question number one, first question was, class 1 restriction enzymes have one of these characteristics. The answer would be that the class 1 enzymes, restriction enzymes, they have ability to cut the DNA as well as modify the DNA by its methylase activity. Question number two was class 2 restriction enzymes have. So class 2 only have nuclease activity. Question number third, a cDNA library will be prepared from DNA, tRNA, mRNA, or rRNA. So the answer is messenger RNA, mRNA. Question number fourth, a genomic library could be prepared from DNA, tRNA, mRNA, rRNA. The answer is DNA. Question number fifth, what is the best system to produce a protein that does not undergo post-translational modification? So the answer is bacteria. The options given to you were bacteria, bacteriophages, yeast, and mammalian cells. So bacteria, they don't have ability to post-translational modification. Although these concepts are changing now. Let me tell you, I just want to, so I try to, when I'm teaching this class, I try to incorporate latest findings as compared to established. There are some concepts which are conventional, but I try to in this course I also include what is the current that is happening. So currently there are some bacteria, there are many bacteria rather which undergo post-translational modification. But for a conventional thinking, yes, but these are very discovery that has happened in last 5 and 10 years with the advanced technology in the field of protein and proteomics. So, but by the conventional thinking, yes, bacteria doesn't have those post-translational modifications like a human cell or a mammalian cells. Question number 6. Oligo-DT will purify which type of RNA? So it will be mRNA because mRNA has poly-A tail, and poly-A tail will complementarize with oligo-DT, which is a DT means a stretch of thymine bases, so that can complementarize with A, and then it can be pulled down. Question number 7: during cloning, which vector from the given below has maximum ability to have largest size of DNA insert? So the answer is cosmid as compared to plasmid and bacteriophages. Question number 8: which enzymes are commonly used in molecular biology? The answer is, the enzyme that is mostly used is class 2 enzymes. Question number 9, which restriction enzyme requires ATP as a cofactor? So the answer would be a class 1 enzyme requires ATP as a cofactor. But I would just go back and look into the slides in case I didn't pick a wrong option. Yeah, class 3 does not require ATP. It's only the class 1 enzyme that requires ATP as a cofactor. Question number 10th, which vector could be used to generate a transgenic plant? So it is TI plasmid. So TI plasmid is a plasmid that can be used to generate recombinant plant. Question number 11, what kind of bonding helps the DNA to become double-stranded from complementary single strand? So hydrogen bond helps the DNA to stay as a double-stranded molecule. Question number 12, a restriction enzyme cuts a DNA fragment by digesting the phosphodiester bond. Question number 13, a cosmid vector is generated by ligating a bacteriophage and a bacterial plasmid. Question number 14, which property of mRNA is utilized for isolation from tRNA and mRNA? So the answer would be 3' poly A tail. But here I have chosen the option E, which says A and D. So although mRNA has unique modifications, or mRNA undergo unique modification at both 5' and 3' end, that is at 5' end it goes capping, and at 3' end it undergoes addition of poly A tail. But for isolating, for extraction of mRNA from a pool of RNA, then we utilized the 3'. So this was a wrong option, wrong answer in the key. It should be only poly A tail. That is at 3' end. Which vector is used to transform insect cells for expressing a heavily glycosylated protein? The answer is baculovirus. Question number 16. If your research is focused on a protein that undergoes post-translational modification, which expression system would be used? So the answer, the options given were bacteria, bacteriophage, a human cell line, E. coli, or all of the above. So the answer is a human cell line. Question number 17. During the process of reverse transcription, which enzyme removes RNA and cDNA from the heteroduplex? The answer is RNase H. Which enzymes removes the DNA loop during the process of reverse transcription? The enzyme is S1 nuclease. A cDNA is generated commonly from which type of RNA? The answer is messenger RNA. Question number 20th. What is the distance in nanometers between two consecutive nucleotides? What is the distance in nanometers between two consecutive nucleotides in DNA? The answer is 0.34 nanometers. Question number 21. How many nucleotides are there in one full turn of B DNA helix? In one full turn of B DNA helices? The answer is 10. What the 22nd question is, what is the distance in nanometer in the full turn of DNA helices? The answer is 3.4 nanometers. Question number 23rd. A phosphodiester bond is formed between carbon atoms. A phosphodiester bond is formed between which carbon atoms in a ribose molecule? The answer would be carbon number 3rd and carbon number 5th. In the DNA double helix, the anti-parallel 5' and 3' strands are due to orientation of carbon in the sugar or ribose molecule. Question number 25th. One second. Question number 25th. The DNA transcribes into mRNA and later mRNA translates into protein. In a particular eukaryotic protein that is 300 amino acids long, what could be the maximum number of nucleotides here? So the answer would be 900 nucleotides. An mRNA with 300 amino acids will have at least 900 nucleotides. Question number 26. Which of the following is not the component of deoxynucleotides that constitute DNA? So the answer is a ribose. A deoxynucleotide will have deoxyribose. Question number 27. Class 2 restriction enzymes have what kind of activity? So class 2 enzymes have only nuclease activity. Which restriction enzymes do not require ATP as a cofactor? So it will be class 3rd and class 2. Classes 3rd and 2 category of enzymes will not require ATP as a cofactor. Again, the repeat question. What kind of bonding helps the DNA to become double-stranded? It's hydrogen bond. Which enzyme is important for removing the phosphate group? The answer is alkaline phosphatase. Question number 31. Promoters are located upstream of the DNA. I repeat, promoters are located upstream of the start site on the mRNA. True or false? So the answer is true. Terminators are located downstream of the start site of mRNA. That's also true. 33rd question. On the mRNA, terminators and promoters code for amino acid, true or false? The answer is false. Question number 34. Do introns code for amino acids? The answer is no. Question 35. Cossack sequence of mRNA is critical for what process? The answer is Cossack sequences could be helpful in translation. Question number 36. Analogous to the phrase copy and paste, which pair of enzyme does the same function? So the answer is copy paste is, or rather the question should be not copy paste, but it should be cut and paste. So this will be enzyme restriction enzymes and ligase. Now, this was a little bit, not well-framed questions. So let me just copy paste those questions here on the whiteboard, and I'll try to explain. I don't know whether I can paste or not. No, I can't paste. So I'll just read out the question. I will read both the questions and I'll tell you what the issue is. So the first question, that is 37th, it says that the blunt end generated by a set of restriction enzyme is referred to as symmetrical or asymmetrical cut, right? So we are talking about blunt. So in these two questions, let me just give you a broad answer. And so symmetrical end, right? Symmetrical, and we are talking about blunt and we're talking about blunt end and cohesive end, right? Blunt end and cohesive end, right? So wherever, so I was reading, so I took a very simplistic concept here. So let's say this is a double-stranded DNA, right? Now, a mechanism that would give rise to a cohesive end will cut here and will cut here. Due to this cutting, what will happen that the fragment generated will be something like this. So the cut happened here, the cut happened here, and the cut happened here. Because of this, you get a cut over here and you get a cut over here. Right? So because this generates this single-stranded here and this single-stranded here. For simplicity, I thought that this would be asymmetrical end compared to the blunt end where the digestion would occur right in the center. So for the sake of simplicity, I made the question that it generates two end, right, which are which are, cut into half. This is like symmetry. And this here that is generated like single strand, this is asymmetric. But in actual, both these cuts, both these cuts, they're happening on a palindromic sequence, because restriction enzyme, they cut palindromic sequence. For example, let's say GATTC, right? Palindromic sequence. You can read from both the side, same sequence. So both these sequences have palindromes. If they have palindromes, then they are symmetrical regardless of blunt end or cohesive end, right? So for both the questions, the answer would be symmetrical. So when I was drafting a question, this question, I mentioned that cohesive will go with asymmetrical and blunt will go with symmetrical. But the right answer is that both of them will go with symmetrical. So that's the problem with the framing of questions, right? We can discuss that later, but let me finish this question. Only 2 or 3 are left. 39th question. Antibiotics listed and below targets the which antibiotic listed below targets the formation of cell wall? The answer is ampicillin. Question number 40th. Movement of DNA during agarose electrophoresis will be from positive to negative, negative to positive. So since DNA is negative charge, it will move from negative to positive. The speed of the DNA movement is proportional to what? So it is inversely proportional to the size of the DNA, right? So with this, we finish the review of quiz 1. Now, the question here is that, there were 2 or 3, so 2 questions. One question was framed wrong. Listen to me very carefully, class. One question was framed wrong. It should have said cut and paste, but I mentioned there copy paste, which is a mistake. Second was that both blunt and cohesive end generating enzymes are symmetrical, but there I made a mistake. So 2 mistakes I made. And so ideally I should be giving you 2 bonus points. But I see there that students have got 41 out of 41, right? So only one student pointed out that there is a confusion with these 2 questions, which is mentioned in the chat area, Tia Foster. So, but none of you have made these points, and many of you, or a few of you have gone 41. So let's think how to grade this quiz, and I'll do what is best for you all, given that there were 2 mistakes from my end. Anybody wants to say anything?
Professor.
Yes.
It's Tia Foster.
Yes.
Shouldn't it be 3 points because you reworded that one wrong, and then the blunt and cohesive ones was 2 separate questions?
Can you slowly speak? I can't hear you.
Can you hear me now?
I can hear you, but speak slowly.
Oh, Okay, I said, shouldn't it be 3 points because one of them was the one you reworded wrong, and then the blunt and cohesive ones was two separate questions.
No, so one of the question was right.
Which one was that?
Let me look.
The
Let
blunt
me
one.
Because it marked it wrong for mine. I had sent you a screenshot.
look.
Okay.
Yeah,
But
the blunt end generates symmetrical cut. That's right.
Yeah, that's symmetrical.
Yeah.
Does it say it's right for me? What does it say?
I can't go and look at your, I'm looking at the questions. I'm not looking at
Oh,
individual grading. I'm
okay.
looking at
So
the questions. So one question out of this and one question was, but the other question is that students have got 41 out of 41. So where do I adjust the bonus? So I get it, 2 points.
So Professor,
So
who those students who didn't get right, you can give them the points, right?
yeah, I'll have to, that's what I said. I have to look into it, what's best for everyone.
Thank you.
But
okay.
if there are ideas, then it's most welcome.
but
So that
Professor.
so what I'm thinking, Nicola, just a second. What
Okay.
I'm thinking that this is happening because of two choices. It's my thinking that this could happen. So let's say true or false question, right? Whether true or false. You have two options, and I'm giving you two attempts, right? So you will pick up one, right? It's natural that okay, I picked up, even if I considering myself as a student, and I get an option to option for two attempts, and then I picked up true, and that is wrong, so I pick up false, right? But the question inherently is wrong, right? So that's one factor. Yes, Nicola, go ahead.
No, I was just thinking about the blunt and the cohesive end. I mean,
Yeah.
in reviewing the notes, I know you mentioned something about being like cohesive end being um like when I mean before I take your quiz, like I reviewed some other stuff, and one was the opposite. One was supposed to be asymmetrical, and one was supposed to be cohesive from the notes.
Yeah, Nicola, that's what I mentioned in middle of my review, that sometimes concept changes as science advances. I talked about this. So, you know, and then you can say that, okay, professor, you know, you are saying this, but I read this now, and so I am taking into the conventional knowledge as well as today's knowledge, right? So I did some homework. So, you know, Tia emailed me, she texted me. So, you know, it's a valid question, right? And I explained my point of view also, why I said asymmetrical, because it gives rise to overhang, which are not of equal length, whereas in blunt end, you get 50-50 chopped off, DNA chopped off from center. So that was my concept of teaching. But you know, if you look into, rather than going into back and forth, now all those sequences which are having palindrome has potential to undergo symmetrical cleavage. And there are exceptions now, right? Because so you can look into, if you go into deep, then you can classify restriction, the action of restriction enzyme on the basis of class 1, class 2, class 3. So class 1 enzymes, they generally do asymmetrical cut, and mm, uh, class 1 does asymmetrical, and class 2 could do symmetrical as well as asymmetrical. So so yeah, that gave me an opportunity to go back and do some homework and look at the latest literature, and so that, you know, I don't want to, what it is, right? And so knowledge is changing very fast, and I cannot drive I can give you the knowledge, I can, but I cannot, you know, make up my things, right, the way I want. So do you get it?
Okay, understood. Thanks.
Yeah. So that's the fact. And many things are developing now. At one point of time, there are only few hundreds restriction enzyme known. Now it is tens and thousands of restriction enzyme known. There's a big biotech company selling thousands of restriction enzymes, and many of them, there are exceptions. So I don't want to be, that, okay, I taught this and this is the fact. I want that when something generates question, it should be analyzed and addressed from all the ends, and knowledge should be the driving force. Are we on one platform? Can I move on?
Professor,
Yes, dear.
the screenshot that I sent you, I don't know why, but it happened to me. I don't know if it happened to anyone else, but when I had put that for the blunt end, it still marked it wrong.
I have no idea. That's a different, going into different direction. I'm just reviewing the question. I don't know what happens to you or what you mark. So I reviewed the question which I gave you, right, dear?
Yeah.
And I accepted what is the norm, what is the current education, and what are the, so I'm being very upfront with that. And what you're ticking, what you're getting, I don't know, but I reviewed the question, right? All right, so let's move on to the next chapter. So the next chapter. So the next chapter would be on mutagenesis. So genesis and recombinant DNA technology. Right, so now these two technology are mainly focused on proteins. So this technology combined, it is also called as protein engineering. Protein engineering. Right? Now, why a protein is being engineered? So protein is being engineered to improve its function, right? Improve the function of proteins. Now, why do you want to improve the function of protein? So function of proteins is improved so that it could have more therapeutic capability, or diagnostic features, more robust therapeutic capabilities, or more um, diagnostic features. So for therapeutic ability, you can think about insulin. Insulin is hormone, which is protein, and given in diabetes. But given that diabetes is another kind of epidemic or pandemic, it is crossing from all the ages now, from young kids to elderly people, and in different parts of the world, right? So So you need insulin which can stand various adverse conditions such as temperature mainly. Increase the half-life of the protein so that it's no more. So if you look at any medicine, you'll see an expiry date, right? That expiry date reflects that activity of that molecule declines, right? So if insulin, let's say in today's date, an insulin protein that is available is good for 1 year, so they want to make it good for 2 years. So it increases the therapeutic potential of insulin. Also, to engineer the molecules in a certain way so that it is more bioactive, right? So it's possible that the first generation proteins which have therapeutic potential could have different limited bioactivity. So increase the biological activity. So in other words, increase the activity and increase the stability. Diagnostic features means, so many of us, we got at our home, a rapid test for COVID, right? So how is this came through mail? It is lying, if it is to be refrigerated, then it goes to fridge. Most of these kits are lying at room temperature. So there must be some very excellent features in these kits because that they can stand room temperature. Most of these kits, they are good at 4 degrees in a refrigerated condition, but they're coming through general mail, which means they're stable in this temperature. So that's why we want to engineer protein. So if we want to engineer protein, right, so you have to understand what protein you are working on, right? If you don't understand what is the nature, because proteins are macromolecules. Proteins are macromolecules that are made up of amino acids, and there are 20 different amino acids, and each amino acid has a carbon atom which is connected to a nitrogen amine, and it has hydrogen also, and oxygen. So these are the two atoms that make up, these are the 4 atoms that make up 20 different amino acids. So some things are very similar in amino acid, and there is one moiety that makes amino acid of 20 different types. So for example, so let's say this is the C alpha. This C alpha is connected to 4 different molecules, right? So this could be hydrogen. Here it could be carboxylic group, and then H2 group, and this will be the 4th would be the R, the side chains. So 20 different amino acids, and the difference is on this R chain, 20 different. It gives rise to 20 different kinds of amino acids. Now, these amino acids can be classified into 4 types depending upon the nature of R: non-polar, polar, acidic, basic, right? I'm not going into the detail now. So 4 different groups depending upon the nature of the R. R is referred as side chains. Now, so you have a protein that could be developed as a therapeutic moiety. So if you see that protein, if you have that protein in your hand, then you have to know the ins and outs of that protein. Otherwise, how can you develop that? So let's go back to the slide and let's talk about one of the proteins. So this is the chapter on mutation. I just want to clarify here that in most of my slides would say in office hours 3 to 4 Monday, but this is slides from a different from a previous semester. This semester your office hours are 4 to 5 on Tuesdays and Thursdays. So your class starts at 4:55 or 5. From 4, the office hour starts. So, so mutagenesis that we understand as change of nucleotide, right? Mutation means change of nucleotide in the DNA. And when nucleotide changes, it can affect a protein because DNA transcribes to RNA and RNA into protein. So if a wrong DNA, then wrong protein, and then wrong function. But now you have to understand that we are not discussing mutagenesis in terms of human diseases, but we are discussing mutagenesis in terms of protein engineering. And we want to make proteins robust so that they become more active biologically. So if they are used for therapy, for treatment, they become more potent. If they are for diagnostics, they become more potent and more robust in picking up diseases. So a mutation is any alteration in the sequence of nucleotides. So just quickly, briefly, mutations can be of several types, but here, let's say if you consider this situation, that there are 3 nucleotides. So you know that 3 nucleotides in mRNA codes for 1 amino acid. So let's say triple U will code for phenylalanine, and UUC will also code for phenylalanine, right? So 2 different triplet code, but they will code for same amino acid. So if U changes to C, yes, it is a mutation, but it has no effect on protein function because both are coding for same amino acid, right? So this would be silent mutation. It has no effect. Now, if you have another mutation where U has been replaced by A, and so you see phenylalanine has been substituted by leucine. These are 2 different amino acids. They have 2 different side chains. So it could have 2 different biological effects, right? So a protein could be tremendously affected by this mutation. So such mutations are missense mutation. It can affect protein function. The third is that this triplet codon that codes for phenylalanine changes into stop codon. So when you change into stop codon, then of course the whole function of a protein is changed, right? So protein could be shorter than what it should be because there is a stop codon, right? So stop codon can totally change the function of a protein. Now, most of these mutations are, if you understand the point mutation, the effect of point mutation, then you can characterize many mutations that can happen. This I teach in detail in my molecular biology class. Now, this is a very interesting protein, right? So let me go back and give you some background about this protein. So in humans, right? In humans, you know that chromosomes, or let's say a single chromosome is made up of DNA and proteins, and proteins which are part of major proteins which are part of chromosome are histone proteins, and these histone proteins are mainly positively charged because these proteins, they form complex with DNA to form nucleosome. Now what happens that there are 4 different kinds of histones: H1, H2A, H2B, H3, H4. 4 or 5 different kinds. There are more subtypes, but let's say in these. Now what happens that if you take histone H3, right? Histone H3 is a protein, right? Now this also has amino acid. Now an amino acid lysine at 27th position undergoes methylation. And once it goes methylation, this leads to gene repression. Gene repression means that a gene that is making up mRNA and protein, a gene that makes up mRNA and protein, whenever required, that's an active gene. But if a gene cannot make mRNA and protein because of methylation on histone H3 lysine number 27, then it can cause gene repression. So this whole biochemical process, methylation of lysine 27 on histone H3, is carried out by an enzyme called as EZH2. Now it has been found that in many cancer, particularly in prostate cancer, EZH2 is defective in prostate cancer. If this is defective, EZH2, then clearly it cannot, if it is defective, then it cannot perform methylation, and hence some of the function of gene expression is affected. So what scientists are doing, they're trying to find a replacement for EZH2, and EZH2 is a big protein. So what happened that scientists found a protein in a smaller eukaryotic organism that is infected by a virus. So interestingly, a virus called as Paramecium Yersinia chlorovirus, right, that infects an algae, it has the same protein that does the function, right? So I repeat again, EZH2 is a protein in human cells that methylates histone H3 that leads to gene repression. If EZH2 is defective, this function is lost and people can develop many kinds of cancer. So if we do some enzyme therapy and replace EZH2 function, not replace, rather support EZH2 function, then maybe we can block the cancer growth. Now, as I mentioned to you, the proteins are made up of amino acid. So how to find out how this protein, where this protein is, where could be this protein is. So one of the sites to look for protein is called as NCBI. So I'm a little bit of bioinformatics I'm introducing here. National Center, NCBI. It's National Center for Biotechnology Information. It is available in public domain. So here, so we can type a name of a protein and it can give us a list of proteins, and we can filter it out, right? Just the way you do your search on Google, you can do this search on Google and you can filter this protein out. Now you get a lot of functions, a lot of information here, but you try to focus on amino acid sequence, right? So one way you can get it quickly is FASTA format. You get only amino acid sequence. Now you want to study this protein as much as possible. So there are various tools that are available that we can study here. So we can look at amino acid content here, right? One thing important that you can look is what is the pH at which this protein could be best soluble if you're working on the lab, in the lab setting. What is the molecular weight, right? How do you look at the secondary structure of these proteins? So there are a lot of bioinformatics tools that we can look into some other class that can help in characterization of a protein. So you understand the protein function, right? So now you have a protein in your hand and you want to understand its function. So one way to understand its function is through inducing mutations. So this is an enzyme that has potential to methylate histone H3 and do same function as EZH2. But we don't know its biochemistry. We don't know how the enzyme function works, right? So if you look at this proteins, this is three-dimensional structure of this protein. So I don't want to go into the details, but through more structure-based technology, you can find out the active site of this protein. Active site is that site that participates in catalysis. Right? So you can get a pocket in this whole protein. You can find a pocket that could be important for catalysis. And here, what is the catalytic property? That this enzyme can methylate lysine 27 on human histone H3. This protein per se is from a virus that infects chlorella, blue-green algae, but it could have human. So this protein can be developed as a therapeutic agent. Now for developing it as a therapeutic agent for cancer treatment, we have to know its function, especially the enzymatic function. Right? So we can, you can introduce various mutations and you can study that which amino acid is participating in the methylation reaction. So this is wild type protein. Each of these band corresponds to enzyme activity. So if you see a band, means enzyme is active. So you put different mutations and you figure out that, long story short, that tyrosine number 105th tyrosine, if you change it to F, Y stands for tyrosine, 105th is the residue number, and F is phenylalanine. If you mutate this, then the enzyme property is lost. So through this knowledge, you study the enzyme kinetics and behavior of this. And then once you study the behavior, then you can develop this as a more potent therapeutic tool. So here, when you see this is wild type and this says Y50A, this means that tyrosine number 50 has been altered to alanine. So at this stage, you should know all the 20 amino acids. You should know all the 3-letter words by which an amino acid can be represented and a single alphabet which represents amino acid. So here tyrosine is 50 if mutated to phenylalanine. There is activity. So if you change this, then you see no activity. So this gives you insight on the function of a protein. So all these are point mutation, a single nucleotide chain, right? And you can study the enzymatic function. This is human histone H3, and this is the V-set protein. When you mix them together, you get a pattern like this. Right? So in next class, we will go into mutations. So more of mutations, right? So one of the mutation could be point mutation where a single nucleotide is changed. Other mutations could be insertional mutations, such that a DNA is inserted. When you insert the DNA, right? When you insert the, so let's say, so let's say, so this is the piece of DNA. Then you can have another piece of DNA, right? This piece of DNA can be inserted here, right? This could be inserted because of, let's say, through restriction enzyme, cut and paste. Right? So for example, but how do you know whether the insertion has happened or not? So let's say you can take an LB, if you have done molecular biology lab, then you can take a bacterial culture plate. So let's say a culture plate would look like this. So if this is the part of bacteria, then you may get, let's say, a purple colony, right, on the LB plate. Now let's say if this insertion happens, then you get here a gene that is now changed. The original gene was this one. Now you have inserted through, let's say, PCR or restriction, digestion, and pasting. Now the effect of this could be that you could get, let's say, you could get a blue colony. This is where you could get a, let's say, a green colony. So this could be the effect of mutations. Or it is possible that you cannot get colony. But what you're trying to understand here, that this portion of the gene, so this is a gene, and this portion of the gene controls the color of the colony, right? So this gene must be making some pigment that is changing the color. Now, this is important, that maybe this is an enzyme with an activity of 10%, and this enzyme has an activity of 80%. So mutagenesis, intentional mutation is, induction of mutation is mutagenesis. So this mutation is being confirmed through this experiment, that okay, mutation is occurring, and through the biological activity, we are coming to know that this activity is increased or not increased. Anything can happen. So what are these? You can see it with eye color, right? The change. So these are phenotypic changes, right? Remember Mendel, he could see the pea plants and make the decision whether it is F1 generation, F2 generation, or what is going on. So phenotypically, you can understand the effect of mutation on a gene in bacteria, because bacteria is easy to work with. But in humans, it is difficult because it could be unethical, right? So, what I did to you in this class, we reviewed the quiz 1 and 2 or 3 questions that were outstanding. We discussed that, and I'll have to see how to, grade or regrade the quiz. And second, we went into learning about proteins, learning about the characteristic of protein, And why do we do protein engineering? And we took one example of histone methyltransferase, which is an enzyme called as EZH2, important for human cancers. It could be defective, and it can lead to aberrant growth of cells. So if we can replace that, or we can provide an extra protein that can mimic the same function of EZH2, then we can be in business of treatment of many cancers. So yeah, so this is what this class was about. And in next class, we will try to finish this chapter. And any questions?
Professor, I just wanted to ask really quickly if you received my emails.
Who is this?
Yael Nakhshumov.
No, I think, did we talk about it last time? I know I haven't received any of your emails.
Yes, homework number 2, I tried to submit and I couldn't, so I wanted to, I sent you an email, and homework number 1 I sent as well in the same email.
No, so let me tell you that I cannot, I'm teaching 3 classes, I cannot receive homeworks by email. There's a place where you have to upload, and each homework has a deadline, right? So if you're not able,
I'm sorry, I wasn't able to upload it on
you have
Blackboard.
to go and talk to Blackboard people, not me. And if, I'm teaching 3 classes, if people keep sending me assignments via email, I'll be totally lost. Sometimes attachment is lost. So I believe I have made this announcement maybe in the beginning of the semester, that please don't submit your assignments by email. It's for you. And assignments, I think assignments contribute to 20%. So work on your Blackboard and talk to Blackboard person in the school why you are not able to upload the assignment. I believe most of the students are able to do assignments, but I didn't receive any. Usually I respond to emails, 99% of them, maybe 1 or 2 falls in the crack, but I haven't received any email from you.
Okay, is it still possible to send it via Blackboard?
The deadline is over.
Okay,
So let's say today. So, but since you were having a hardship, I'll give you a chance that with this 3rd assignment, so for this chapter, you submit your 1st and 2nd assignment together. After that, then there is no grace.
Okay, thank you.
All right. Yeah.
Professor?
Yes, Tatiana.
Did you get my name for the attendance? I had came like a few minutes late.
Okay, let me see. So I saw Kyla Adetji. Are you here?
Yes, hi, Professor. I'm here. I just wanted to make sure you got my attendance as well.
Okay.
Yes. Do you have my attendance?
Yeah, I'm trying to work. Something is wrong with my Excel. So I'll have to, I'll put it there.
Yes. Just making sure.
Okay. 2 participants raised hand. Yes. So one person is 917412. Just write your name on the chat or write your question.
Professor, 917412 is Sandra. You already got me.
Okay, good. Yeah, I got you.
Professor, did you get
you
me on
got
attendance?
me, right?
I can hear to only one person. Go ahead, tell me.
Tatiana Ross.
Just a second. I'm having something with my Blackboard. Not Blackboard, but Excel sheet. Tatiana Ross. Highlighted. Anybody else?
يعل نخشمة.
Okay. All right. Kyra Donald, okay. All right, if there are no more questions, then we can end this class.